Welcome to Colunk Igniting the Curious Mind has on date 24 June 2026. Principle of Inheritance and Variations. Have you ever wondered why an elephant always gave birth to only to baby elephants, not some other animals? Or why a mango seeds always form mango plants and not any other plants, given they do are the offsprings identical to their parents and or they show a differences in some of their characteristics? Have you ever wondered why some siblings sometimes look similar to each other or sometimes even so different? These are several related questions that are dealt with scientifically in a branch of biology known as genetics. This subject deals with the inheritance as well as that of variations of characters from parents to offsprings. Inheritance is a process by which characters are passed on from parents to progeny. It is a basis of heredity. Variation in the degree by which a progeny differ from their parents. Human knew from the early as 8000 to 1000 BC that one of the causes of variations was hidden in the sexual reproductions. They exploited the variations and were naturally present in the wild population of plants and animals, selectively breeded and selected from organisms that possess desirable character. For example, through artificial selections, domestication from ancestral wild cow, as we well know Indian breeds. The Shaiwala cow in Punjab, we must note, however, recognizing that our ancestral knew about inheritance of character and variations, they had a very little idea about scientific basis of these phenomena. Mendel's law of inheritance. It was during the mid-19th century that headway was made in understanding the inheritance. George Mendel conducted a hybridization experiment on the ground peas for 7 years, 1956 to 1963, and proposed a law of inheritance in the living organism. During Mendel's investigations into inheritance patterns, it was the first time that the statistical analysis and mathematical logics were applied to the problems in biology. His experiment had a large sampling size, which gave a great credibility to the data that he collected. Also, confirmation of this inferences from the experiments on successive generations of his test plants provided that these results pointed to a general rule of inheritance rather than being substantiated idea. Mendel's investigated characters in the garden pea plant that were manifested as two opposite traits, tall and dwarf plants, yellow and green seeds. This allowed him to set the basic frameworks of the rules of governing inheritance, which was expanded on later scientific to account for all the diverse natural observations and the complexity of inheritance in them. Mendel conducted about such artificial cross-pollination experiment using several true breeding lines. A true breeding line is one that having undergone a continuous self-pollinations and shows a stable trait of inheritance and expressions of several generations. Mendel selected 14 breeding pea plants varieties as pairs which were similar except one character with the contrasting traits. Some of the contrasting traits selected were smooth or wrinkled seeds, yellow or green seeds, inflated or a constricted green or yellow pods, tall or dwarf plants. Inheritance of one gene. Let us take the example of one such hybridization experiment carried out by Mendel, where he crossed a tall dwarf pea plant to study the inheritance of one gene. He collected seed produced as a result of cross-grew them and generated plants of the first hybrid generations. This generation is called as filial progeny or F1. The Mendel observed that all the F1 progeny of the plants were tall like one of its parents, none were dwarf. He made similar observation for the other pairs of the trait. He found that F1 always resembled either one of the parent and the trait of the other parent were not seen in them. Mendel also then self-pollinated the F1 plants and to surprise found that it was a F2 generation filial. Two generations of some of the offsprings were dwarf. The character that one not seen in the F1 generation was now expressed. The proportion of plants that were dwarf were 1/4 of the F2 plants, while the 3/4 of the F2 plants were tall. The tall and dwarf traits were identical to the parental type and did not show any breeding. Blending. This is all the all the offsprings were either tall or dwarf. None were in between the heights. Similar results were obtained while the other traits that he studied. Only one of the parental trait was expressed in F1 generation, while at the F2 stage, both the traits were expressed in a proportions of 3:1. The contrasting traits did not show any blending at either F1 or F2 stage. Based on the observation, Mendel proposed that something was being stably passed down unchanged from parent to offspring to the gametes. The successive generation he called as factors. He called these as factors. Now we call them as genes. Genes therefore are the unit of inheritance. They contains information that is required to express particular trait in an organism. Genes which code for a pair of contrasting traits are known as alleles. They are slightly different from the same alleles. If we use alphabet symbols for these alleles, then the capital letter is used for the trait expressed at F1 stage and the small alphabets of the other traits. For example, in case of a characteristics of a height, capital T is used and for the taller trait and small t is used for the dwarf. Capital T and small t are the alleles of each other. Hence, in a plants with a pair of alleles for the height would be capital T, capital T, small t and small t. Mendel also proposed that in a true breeding, the tall and dwarf pea plant variety and the allelic pairs of genes for a height are identical and homogeneous. The capital T, small t respectively. The T capital and small t are also called as genotype of the plant, while the descriptive term is tall and dwarf are the phenotype. That when of the phenotype of a plant could had a genotype capital T small t. Has a Mendel found that phenotypic of the F1 heterozygotes capital T small t to be exactly like the capital T parent in appearance. He proposed that in pair of a dissimilar factor, one dominates the other has an F1 and hence he called the dominant factor while the other factor is recessive. In this case, capital T for tallness is dominant over small t dwarfness, that is recessive. He observed that identical behavior for all the other character traits pair of he studied. It is convenient and logical to use a capital and a lowercase as an alphabetic symbol to remember this concept of dominance and recessiveness. Do not use T for tall and D for dwarf because you will find it difficult to remember whether capital T or small d are the allele of the same gene or the characters or not. The allele can be similar as in case of homozygotes of capital T, small t or can be dissimilar as in case of heterozygote capital T small t. Since the capital T small t plant is a heterozygous gene controlling one character height and it is monohybrid, and the cross between capital T small t is a monohybrid cross, from the observation the recessive parental trait is expressed without any blending in F2 generation. We can infer that when a tall and a dwarf plant produce gametes, but the process of meiosis, alleles of a parental pair separates and segregates from each other and only one allele is transmitted to a gametes. This segregation of allele is a random process, so there is 50% of chances of gametes containing either allele has been verified by the results of crossing. In this way, the gametes of a tall capital T plants have an allele capital T and the gametes of dwarf small t plants have the allele T. During fertilization, the two alleles T and from the one parent and through the pollen and the small t from the other parent when through the egg are united to produce united to produce these hybrids. Since these hybrids contain alleles which express contrasting traits, the plants are heterozygous. The production of gametes by the parents and the formation of zygotes. The F1 and F2 plants can be understood from a diagram called Punnett square shown in the figure. It was developed by a British genetics Leonard C. Punnett. It was a graphical representation to calculate the probability of all possible genotypic of offspring in a genetic crosses. The possible gametes are written in two sides, usually the top rows and the left column. All possible combinations are represented in the box below the squares, which generates a square output. The Punnett square shows a parental tall TT, male dwarf small t, female plants. The gametes produced by them and the F1 capital T small t progeny. The F1 plants of a genotype capital T small t are self-pollinated. The symbols as shown here are used to denote the female and the male pollen, the F1 generation. That represents respectively the F1 plants of a genotype capital T small t, when self-pollinated, produces a gametes of a genotype cap T and small t in an equal proportion. When the fertilization takes place, the pollen grain of the genotype T have a 50% of chances to pollinate the eggs of the genotype T capital T as well as of genotype small t. Also, pollen grain of genotype T has 50% of chance to pollinate egg of genotype capital T as well as a genotype small t. As a result, random fertilization resultant zygotes can be genotype capital T, capital T, capital T, small t, small t, small t. From the Punnett square, it is easily seen that 1/4 of the random fertilization leads to capital T, small capital T, capital T, half leads to capital T, small t, 1/4 to small t, small t. Though the F1 have a genotype of capital T, small t, but the phenotypic character is seen to be tall, at F2, 3/4 of the plants are tall, whereas some of the plants are TT while the others are capital T small t. Externally, it's not possible to distinguish between the plants with the genotype of capital T small t. Hence, within the genotype ratio capital T small t, only one of the character tall is expressed. Hence, the character capital T or the tall is said to dominate over the other allele T or the dwarf character. It is this due to the dominance of one character over the other. At all the F1 are tall, and in F2, 3/4 of the plants are tall. Genotypically, 1/2 of the capital T small t, 1/4 are capital T. The slates of phenotypic ratio of 3/4 of the tall ratio, that is 3:1 ratio, but the genotypic ratio is 1:2:1. 1/4 is to 1:2 is to 1/4 ratio. Capital T, small t is a mathematical condensable to form a binomial expression ax plus by whole square that has a gametes that bear in the gene capital T small t equal to the frequency of half. This expression is expanded as given below. Mendel self-pollinated the F2 plants and found that the dwarf F2 plants continued to generate a dwarf plant in F3, F4 generation. He concluded that the genotype of the dwarf was homozygous, capital T, small t. What do you think he would have got when he had self-pollinated capital tall F2 plants? From the preceding paragraph, it's clear that through the genotype ratio can be calculated using mathematical probability by a simple looking at a genotype of a dominant trait. It's not possible for us to know the genotype composition. That is, for example, whether a tall plant from F1 or F2 has a capital T, small t composition cannot be predicted. Therefore, to determine the genotype of a tall plant at F2, Mendel crossed the tall plant from F2 with a dwarf plant, and he called as test cross. In a typical test cross, an organisms showing a dominant phenotype and those of the genotype is to be determined is crossed with a recessive parent instead of self-crossing. The progeny of such cross can be easily analyzed to predict the genotype of the testing organism. This shows the results of typical test cross where violet color flower is dominant over the white color. Using Punnett square, try to find out the nature of offspring of test cross. What ratio did you get using the genotype of this cross? Can you give the general definition of a test cross? Based on observations of monohybrid crosses, Mendel proposed two general rules to consolidate this understanding of inheritance in the monohybrid crosses. Today, the rules are called as principle of law of inheritance. The first law or the law of dominance, and the second law or the law of segregations. Law of dominance: characters are controlled by discrete unit called as factors. Factors occurs in pair. In a dissimilar pair, the factors one member of the pair dominates the other, dominant and recessive. The law of dominance is used to explain the expressions of only one of the parental character. In a monohybrid cross, the F1 and the expressions of both F2, it has explained the proportions of 3:1 obtained at the F2. Law of segregation. This law is proposed by the fact that the allele do not show any blending and that both the characters are recovered such in the F2 generation through one of these are not seen in F1 generation. The one parent contains two alleles during the gamete formation. The factor or the allele of the pair segregates from each other such that the gamete receives only one factor. Of course, the homozygous parents produces all the gametes that are similar, while the heterozygous one produces two. Incomplete dominance. When an experiment on P was repeated using other traits in other plants, it was found that sometime F1 had a phenotype that did not resemble either of the two parents and was in between the two. The two parents was in between the two. The inheritance of the flower color in the dog flower snapdragon or the Anthrachinus nump species, a good example to understand incomplete dominance. In a cross between the true breed red flower RR and the true breeding white flower plants, the F1 capital R small r was the pink. When the F1 was self-pollinated, the F2 resulted in the following ratio: RR red and 2 pink, 1 white, where the genotypic ratio was exactly as we would expect in any other Mendelian monohybrid cross, but the phenotype ratio had changed from 3:1 dominant recessive. What happened was R was not completely dominant over R, and this made it possible to distinguish capital R small r as a pink from RR and R small r white. Explanation of the concept of dominant. What exactly is dominance? Why are some alleles are dominant and some recessive? To tackle this question, we must understand what a gene does. Every gene, as you know by now, contains the information to express particular traits. In diploid organism, there are two copies of each of the gene has pairs of allele. Now, these two alleles need not always be identical as in the heterozygote. One of them may be different, but due to changes it had been undergone, about which you will read further and in next chapter, which modifies information that particular allele contain. Let us take an example of a gene that contains informations for producing enzymes. And now there are two copies of these gene, the two allelic forms. Let us assume, as is more common, that the normal allele produce the normal enzyme that is needed for the transformations of substrate. Yes. Theoretically, the modified allele could be responsible for production of normal, a less efficient enzyme, a functional enzyme, and no enzyme at all. In the first case, the modified allele is equivalent to the unmodified allele, that is, it will produce the same phenotype trait, that is, results in the transformation of substrates. Yes, such equivalent allele pairs are very common, but in the allele produces a non-functional enzyme or no enzyme. the phenotypic way will be affected. The phenotype trait will only be dependent on the functioning of the unmodified allele. The unmodified functioning allele, which represents the original phenotype, is a dominant allele, and modified allele is generally the recessive allele. Hence, in the example above, recessive trait is seen due to non-functional enzyme or because no enzyme is produced. Codominance. Till now, we have discussed crosses where the F1 resembles either of the two parents, dominance, or was in between, incomplete dominance. But in case of codominance, the F1 generation resembles both parents. A good example is different types of red blood cells that determine ABO blood grouping in human being. ABO blood group blood are controlled by gene I. The plasma membrane of red blood cells has a sugar polymer that protrude from its surface, and the kind of sugar is controlled by the gene. The gene I has three allele IAIBI, and allele IAIB produces slightly different form of sugar while I does not produce any sugar. Because humans are diploid organism, each person possesses I, in other words, where I are present only IA expressed. Because I does not produce any sugar. When IB and I are present, IB expressed, but IA and IB are present together, they both express their own types of sugar. This is because the codominance. Hence, red blood cells have both I, A and B types of sugar. Since there are three different alleles, there are 6 different combinations of these 3 alleles that are possible, and therefore total 6 different phenotype of a human ABO blood type. How many phenotypes are possible? Now, do you realize that the examples of ABO blood grouping is also provides a good example of multiple allele? Here you can see that there are more than three allele governing the same character. Since in an individual only 2 allele can be present, multiple allele can be found only when the population studied are made. Occasionally, a single gene product may produce more than one effect. For example, a starch synthesis in pea seed is controlled by one gene. It has 2 allele, B and B. Starch synthesized directly by BB or homozygote, therefore, large starch grains are produced. In contrast to small letter b and B, homozygotes have lesser efficiency in starch synthesis, produce a smaller starch grain. After maturations of the seed, the BB seeds are found and small letter bb seeds are wrinkled. The heterozygote produced ground seeds, and so B seems to be a dominant allele, but the starch grain produced are intermediate size, capital B, small me seeds. So if the starch grain seed size is considered as a phenotype, then from this angle, the allele shows incomplete dominance. Therefore, a dominance is not autonomous feature of a gene or the product that it has information for. It depends on the gene's product and the production of particular phenotype from the product. As it does not a particular phenotype that we choose to examine in case more than one phenotype is influenced by the same gene. Inheritance of 2 gene. Mendel has also worked with the uncrossed pea plant that differed into character, has seen in the cross between a pea plant that seeds with a yellow color and a round shape, and one that had seeds of green color and wrinkled shape. Mendel found that the seeds resulting from the crossing of parents had a yellow colored and round shaped seeds. Here you can tell which of the character in the pair yellow, pair green, and the round wrinkled shape was dominant. Thus, yellow color was dominant over green and round shape dominant over wrinkled. These results were identical to those we got when we had separate monohybrid crosses between yellow and green seeded plants and between round and wrinkled seed plant. Let us now see the genotype Y, capital Y for a dominant yellow seeds color and a small y for recessive green seed color, capital R round shaped seeds and small letter r for the wrinkled seed shape. The genotype of the parents can be written as a RRYY, small r, small yy. The cross between the two plants can be written down in the figure shown. Genotype of the parent plant. The parent, capital R, capital Y, small y, unite on fertilization to produce F1 hybrid, capital R, capital Y, small y. When Mendel self-hybridized an F1 plant, he found that 3/4 of the F2 plants had yellow seeds and 1/4 had green. The yellow and green colors segregated in a ratio 3:1. Round and wrinkled seed shapes also generated in a 3:1 ratio, just like a monohybrid cross. Law of Independent Assortment. In a dihybrid crosses, the phenotype round, yellow, round, green, wrinkled, green appeared in the ratio 9:3:3:1. Such a ratio was observed for several pairs of character that Mendel studied. The ratio of 9:3:3:1 can be derived as a combination of series of 3 yellow, 1 green and 3 round and is to 1 wrinkled. This derivation can be written as 3 round is to 1 wrinkled, 3 yellow, 1 round equals to 9, yellow is to 3 wrinkled yellow is to 3 round green is to 1 wrinkled green. Based on the observation of dihybrid cross between plants differ in two traits. Mendel proposed a second set of generalization that we call Mendel's law of independent assortment. The law states that when two pairs of traits are combined in a hybrid, segregating of one pair of character is independent of the other pair. The Punnett square can be effectively used to understand the independent segregations of two pairs of gene during meiosis and produce production of egg and pollen in a F1 capital R, capital Y, small y plant. Considering the segregation of one pair of gene R and small r, 50% of the gametes have gene R. The 50% of small r. Besides each of gametes having either capital R, small r, it should also have an allele of capital Y, small y. The important thing to remember here is the segregation of 50% of R and 50% of small r is independent from the segregation of 50% of capital R, 50% of Y. Therefore, 50% of the R bearing gametes has Y, capital Y, and 50% has small y. Similarly, 50% of capital R bearing gametes has capital Y and other 50% has a small phi. Thus, there are 4 genotype of the gametes, 4 types of pollen and 4 types of egg. The 4 types of capital R small y, capital R small y, are capital R small, capital Y small, R small y, each having a frequency of 25% or 1/4 of the total gamete produced. When you write down the 4 types of egg and pollen on the two sides of Punnett square, it's very easy to derive the composition of zygotes that give rise to F2 plants. Although there are 16 squares, how many different types of genotypes and phenotypes are formed? Now, can you, using the Punnett square data, work out the genotypic ratio at F2 stage? Fill in the format given. In the genotypic ratio, also 9:3:3:1. Chromosomal theory of inheritance. Mendel published his work of inheritance character in 1865, but for several reasons it remained unrecognized till 1900. Firstly, communication was not easy as it's now. Now, in those days, his work could be not be widely published. Secondly, his concept of gene factor in Mendel's word as stable, discrete unit that controlled the expression of traits of the pairs of allele which did not blend with each other was not accepted by the contemporaries as an explanation for the apparently continuous variations in nature. Thirdly, Mendel approach used mathematics to explain the biological phenomena was totally new and unacceptable to many of the biologists of his time. Finally, though Mendel's work suggested that the factor gene were discrete unit, he could only not provide in physical proof for the existence of a factor or say that they were made up of. In 1900, 3 scientists, Dihybrid, Corin, and von Tassmacher, independently rediscovered Mendel's results of inheritance of character. Also, by this time, due to advancement of microscope that were taking place, scientists were able to carefully observe all the cells. This led to discovery of the structures of nucleus that appeared to be double and divided to just before the cell division. This was called as chromosomes. A colored body has were visible by the staining. In 1902, chromosome movement during meiosis had been worked out. Walter Sinton and the Theodore Bauer noted that the behavior of the chromosome was parallel to the behavior of the gene, used chromosomal movement to explain Mendel's law. Recalling what you have studied of the behavior of chromosome during mitosis, equational divisions, during meiosis, reductional divisions, the important thing to remember of the chromosome as well as the gene occur in pair. Two alleles of a gene pairs are located in a homologous sites of homologous chromosomes. During the anaphase of meiosis 1, the two chromosome pair can align at the metaphase plate independent of each other. To understand this, compare the chromosome of 4 different color in left and right column. In the left column, possibility orange and green are segregated together, but in the right column, possibility 2, the orange color chromosome is segregated with the red chromosomes. The Sutton and Bowyer argued that the pairing and the separation of pair of chromosome would lead to the segregation of pair of factor they carried. The Sutton united the knowledge of chromosomal segregation with Mendel's principle and called it as chromosomal theory of inheritance. Following this synthesis of idea, experimental verification, the chromosome theory of inheritance by Thomas Hunt Morgan, his colleagues, led to a discovery of basics of variation that sexual reproduction produced. Morgan worked with the tiny fruit flies, Drosophila melanogaster, which were found very suitable for such studies. They could be grown on simple synthetic media in laboratory. They complete their life cycle in about 2 weeks, and a single mating could produce a large number of progeny flies. Also, there was a clear differentiation of sexes. Male and the female flies are easily distinguishable. Also, it has many types of heredity variations that can be seen with a low power microscope. Linkage and recombination. Morgan carried out several dihybrid crosses in Drosophila to study genes that were sex-linked. The crosses were similar to dihybrid crosses carried out by Mendel in the pea. For example, Morgan dihybrid yellow bodies, white-eyed female to a brown body, a red-eyed male, and intercrosses their F1 progeny. He observed that the two genes did not segregate independently of each other, and F2 ratio derivatives vary very significantly from 9:3:1 ratio, when the two genes are independent. Morgan and his group knew that the genes were located on X chromosome and saw quickly when the two genes in a dihybrid cross were situated on the same chromosome. The proportion of the parental gene combinations were much higher than the non-parental type. Morgan attributes this due to the physical association or linkage of two genes, coined with the term linkage to describe as physical associations with a gene or chromosome, and the term recombination to describe the generations of non-parental gene combinations. The Morgan and his groups also found that even when the genes were grouped on the same chromosome, some genes were tightly linked, show a very low recombinations, while the other were closely linked, showed a high recombinations. For example, he found that the gene white and yellow were tightly linked, showed that only 1:3% recombination, white and miniature wing. Then 37:2% recombination. His student Alfred Stervent used a frequent recombinations between the gene pair on the same chromosome as a measure to distance between the gene and mapped their positions on the chromosome. Today, genetic map are extensively used to starting point in the sequence of a whole genome, as was done in the case of human genome sequence project described later. Polygenic inheritance. Mendel studied mainly described those traits that were distinct alternate forms, such as flower color, which were either purple or white. But if you look from around, you'll find that there are many traits which are not so distinct in their occurrence and the spread across a gradient. For example, in human, we also just have tall and short people has two distinct alternative but whole range of possible heights. Such traits are generally controlled by three or more genes and are thus called polygenic traits. Besides the involvement of multiple gene, allelic inheritance also takes into account influence of environment. Human skin color is another classic example for this. In a polygenic trait, the phenotype reflects the contribution of each allele. The effect of each allele is additive. To understand this better, let us assume that the three gene ABC control the skin color of a human, with the dominant A, B, and C responsible for dark skin color, the recessive form ABC, a light skin color, and all the genotype with all the dominant allele A, B, B, C, C will give a darkest skin color, and with all the recessive allele, that is small a b b c c will have a lighter skin color. As expected, the genotype with the three dominant allele and the three recessive allele will have an intermediate skin color, and in this manner the number of each type of allele in a genotype would determine the darkness and the lightness of a skin in an individual. Philanthropy. We have so far seen the effect of gene on a single phenotype or a trait. There are, however, instances where single gene can exhibit multiple phenotypic expressions, such as gene is called as pleiotropy gene. The underlying mechanism of pleiotropy in most cases is the effect of gene on the metabolic pathway, which contributes towards a different phenotypic. For example, diseases of phenylketonuria, which occurs in a human. The disease is caused by a multiplication in the mutation in the gene that codes for the enzyme phenylalanine hydroxylase, single gene mutation. This manifests itself through a phenotypic expression characterized by mental retardation and reduction in the hair and skin pigment. Sex determination. The mechanism of sex determination has always been a puzzle before the genetics. The initial clue about the genetic chromosomal mechanism of sex determination can be traced back to some of the experiment carried out in the insect. In fact, cytological observation made in a number of insects led to a development of a concept, genetic chromosomal basis sex determination. Henklin, 1891, could trace a specific nuclear structure all through the spermatogenesis in a few insects. It was also observed that by him that 90% of the sperm received his structure after the spermatogenesis, whereas 50% sperm did not receive it. Henklin gave a name for this structure as X-body, but he could not explain the significance. Further investigation by other scientists led to a conclusion that X-body of the Henklin was in fact a chromosome, and that is why it was given the name X chromosome. It was also observed that a large number of insect mechanism of sex determination is a exotype, all egg bearing additional X chromosome besides the other chromosome autosomes. On the other hand, some of the sperm bear X chromosome, whereas some do not. Eggs fertilized by the sperms having an X chromosome becomes female, and those fertilized by sperm did not have an X chromosome became male. Do you think the number of chromosomes in the male and females are equal? Due to the involvement of X chromosome in the determination of a sex, it was designated to be a sex chromosome, and the rest was the chromosome were named as autosomes. The grasshopper is an example of an exotype of sex determination in which the males have only one X chromosome besides autosomes, whereas a female have a pair of X chromosome. This observation leads to the investigations of number of species to understand the mechanism of sex determination. In a number of other insects and mammals, including man, XY types of sex determination is seen where both male and females have same number of chromosome. Among the males have X chromosome is present only, but its counterpart is distantly smaller and called Y chromosome. The female, however, has a pair of X chromosomes. Both male and female bears the same number of autosomes. Hence, male have autosome plus XY, while female have autosome XX. In human being and in the Drosophila, the male have one X and Y chromosome, whereas a female pair have X chromosome besides autosomes. In the above description, you have studied about the two types of sex determination mechanism, exotype, XY type. But in both the cases, male produce two different type of gametes, either with or without X chromosome, or some gametes with X chromosome or with Y chromosome. Such type of sex determination mechanism is designated to be an example of male heterogamety. In some other organisms, births are for different mechanisms sex determination is observed. In this case, total number of chromosomes is same as in both male and female. Two different types of gamete in terms of sex chromosomes are produced by the female heterogamety. In order to have a distinctions with the mechanism of sex determination described earlier, two different sex chromosomes of the female bird has been designated to be the Z and W chromosome. In these organisms, the female have one Z and one W chromosome, whereas a male have a pair of Z chromosome besides autosomes. Sex determination in human. It was already been mentioned that the sex determination mechanism in the case of humans is XY type. Out of 23 pair of chromosome present, 22 are exactly same in both male and female. These are autosomes. A pair of X chromosomes are present in female, whereas the presence of X and Y chromosomes are determinant of the male. The gametes are produced. 50% of total sperm produced carry the X chromosome, and the rest 50% has Y chromosome besides autosomes. Female, however, produce only one type of ovum with the X chromosome. There is an equal probability of fertilizations of ovum with the sperm carrying their either X and Y chromosome. In some cases, ovum fertilized with the sperm carrying X chromosome, the zygote developed into a female XX, and fertilized ovum is Y chromosome carrying out the sperm results into a male offspring. Thus, it is evident that it is the genetic makeup of the sperm that determines the sex of a child. It is also evident that in case of pregnancy, there is 50% of probability of either a male or a female child. It is unfortunate in our society are blamed for giving birth to a female child and being ostracized as and ill-treated because of the false notions. Sex determination in honeybee. Sex determination in honeybee is based on the number of set of chromosomes an individual receives. An offspring formed from a union of sperm and an egg develops as a female queen or worker, and unfertilized eggs developed as a male drone by a means of parthenogenesis. This means that the male have half the number of chromosomes than that of a female, are diploid having 32 chromosomes, and the males are haploid having 16 chromosomes. It's called as haplodiploid sex determination system that has a special characteristic features. Has a male produce sperm by mitosis. They do not have father, thus cannot have a son, but have a grandfather and can have a grandson. How is the sex determination mechanism different in the birds? In the sperm or the egg responsible for the sex of a chick's mutation. Mutation is a phenomena which results in alternation of DNA sequence, and the consequently results in a change in the genotype and the phenotype of an organism. In addition to recombination, mutation is another phenomena that leads to variations in DNA. You have learned in chapter 5 DNA helix runs continuously from the one end to the other end in a chromatids. It's highly supercoiled form, therefore allows deletions or again insertion or duplication is the segments of a DNA. Results in alternation of chromosome. Since genes are known to be located on the chromosome, alternation in the chromosome results in the abnormalities and aberrations. Chromosomal aberrations are commonly observed in the cancer cells. In addition to the above, the mutation also rises due to change in the single base pair of DNA. This is known as a point mutation. A classical example of such mutation is a sickle cell anemia. Deletions and insertion of base pairs of DNA causes a frameshift mutations. The mechanism of mutation is beyond the scope of these discussions at this level. However, there are many chemicals and physical factors that induce mutations. These are referred to as mutagens. UV radiations can cause mutation in the organism. It is mutagens. The genetic disorder. Pedigree analysis. The idea that disorder are inherited and have been prevailing in the human society since long. This was based on the heritability of a certain characteristics feature in the family. After rediscovering of Mendel's work and practice of analysis, inheritance patterns of traits in human being began. Since it is evident that the controlled cross can be performed in a pea plant or the same organism, not possible in case of human beings. Study of family history about inheritance is a particular trait provides an alternative. Such an analysis of trait on several generations of families is called as pedigree analysis. In the pedigree analysis, inheritance of a particular trait is represented in the family tree over a generations. In human being, pedigree study provides a strong tool which is utilized to trace the inheritance of specific trait, abnormality or the disease. Some of the important and standard symbols used in the pedigree analysis is shown in the figure. As you have studied in this chapter, each and every feature in any organism is controlled by one or the other gene located on the DNA represented in the chromosome. DNA is carrier of genetic information. It is hence transmitted from one generation to another without any chance or change or alternations. However, changes or the alternation do not take occasionally. Such an alternations or changes in genetic material is referred to as mutations. A number of disorders in human beings have been found to be associated with the inheritance of changed and altered gene or the chromosome. Broadly, genetic disorder. Mendelian disorder. Mendelian disorder, broadly, genetic disorder may be grouped into two categories: Mendelian disorder, chromosomal disorder. Mendelian disorders are mainly determined by the alternation and mutations of in the single gene. These disorders are transmitted to the offspring on the same lines, as well as studied in the principle of inheritance. The pattern of inheritance of such Mendelian disorder can be traced in a family by a pedigree analysis. Most common and prevalent Mendelian disorders are Haemophilia, cystic fibrosis, sickle cell anemia, color blindness, phenylketonuria, thalassemia, etc. It's important to mention here that such Mendelian disorder may be dominant or recessive. By the pedigree analysis, one can easily understand whether the trait is in the question is dominant or recessive. Similarly, the trait may also be linked to a sex chromosome, as in case of hemophilia. It is evident that the X-linked recessive trait show a transmission from carrier female to male progeny. A representative pedigree is shown for dominant and a recessive trait. Discuss with your teacher to design the pedigree chart linked to both autosomes and sex chromosome. Color blindness. It is sex-linked recessive disorder due to the defect in the red color cone of the eye, resulting in the failure to discriminate between the red and green color. This defect is due to mutation in certain gene present in X chromosome. It occurs in 80% of males and only 0.4% in female. This is because the gene is led by the red-green color blindness are on the X chromosome. The males have only X chromosome and the females have 2. The son of the woman who carries the gene have 50% chance of being colorblind. The mother is not herself colorblind because the gene is recessive. This means that the effect is suppressed by her matching dominant normal gene. A daughter will not normally be colorblind, but unless her mother is carrier, her father is a colorblind. Hemophilia. It's a sex-linked recessive disease which shows its transmissions from unaffected carrier female to the male progeny. It has been widely studied in this disease. The single protein that is a part of the cascade of proteins involved in the clotting of the blood is affected. Due to this, it is in an affected individual, a simple cut can result in a small non-stop bleeding. Heterozygous female carrier for a hemophilia hemophilia may transmit a disease to son. The possibility of female becoming a hemophilic is extremely rare there because mother of such female has to be at least a carrier and the father should be hemophilic, unviable in the later stage of life. Family pedigree Queen Victoria shows a number of hemophilic descendants as she was a carrier of the disease. Sickle cell anemia. It's an autosome linked recessive trait that can be transmitted from parents to offspring when both the parent are the carrier for the gene heterozygous. The disease is controlled by a single pair of allele HA-B-HBS. Out of the three possible genotype, only the homozygous individual of HBS, HBS shows a disease phenotype. Heterozygous HBS individuals appear apparently unaffected, but they are the carrier of the diseases with 50% probability of transmission of the mutation, the gene of the progeny. Thus exhibits a sickle cell trait. The defect is caused by the substitutions of glutamic acids by the valid at the sixth position of beta globin chain of the hemoglobin molecule. The substitutions of amino acids in the globin protein results due to the single base substitutions at a sixth codon of the beta globin and CAG to GAG to GUG. The mutant of hemoglobin molecule undergo a polymerization due to low energy tensions, causing the change in the shapes of RBC from a biconcave disc to an elongated sickle-like structure. Phenylketonuria. This is an inborn error of metabolism and is also inherited as an autosomal recessive trait. The affected individual lack an enzyme that convert amino acid phenylalanine into a triosine. As a result, phenylalanine is accumulated and is converted into a phenyl pyruvic acid and their derivatives. Accumulation of these in the brain results in the mental retardations. These excreted to the urine becomes its poor absorption by the kidney. Thalassemia. This is also an autosomal linked recessive blood disease transmitted from parent to offspring when both the parents are unaffected carrier of the further gene or the heterozygous. The defect could be due to either the mutation or deletions, which ultimately results in a reduced rate of synthesis of one of the globin chain, alpha and beta chain, that makes up the hemoglobin. This causes a formation of abnormal hemoglobin molecule, resulting into anemia, which is a characteristic of the disease. The thalassemia can be classified according to which the chain of hemoglobin molecule is affected. Alpha thalassemia, production of alpha globin chain is affected with the beta thalassemia, production of beta globin chain is affected. Alpha thalassemia is controlled by two closely linked HBA1 and HBA2 on the chromosome 16 of the parent, and it's observed due to the mutations or deletion of one or more 4 genes. The more gene affected, the less alpha globin molecule produced, while the beta thalassemia is controlled by a single gene HBB on the chromosome level of each parental and can occur due to mutations of one or more gene. Thalassemia differ from sickle cell anemia in that form is a qualitatively problem by synthesis of 2 new globin molecules, while the later is qualitative problem of synthesis of incorrectly functioning globin. Chromosome disorder. The chromosome disorder is, on the other hand, are caused due to the absence of or excess abnormal arrangement of one or more chromosome. Failure of segregation of chromatids during cell division cycle results in the gain or loss of chromosomes called aneuploidy. For example, the Down syndrome results in the gain of extra chromosome 21. Similarly, Turner syndrome results in the loss of X chromosome in the human female. Failure of cytokinesis after telophase stage of cell divisions results in an increase in the whole set of chromosome in an organisms, and this phenomenon is known as polyploidy. This condition is often seen in the plants. After the total number of chromosomes in the normal human is 46 pair, 46, that is 23 pair, out of which 22 pairs are autosomes and 1 pair of chromosome of sex chromosome. Sometimes, rarely, either the additional copy of chromosomes or may be included in an individual. An individual may lack one pair of chromosome. These situations are known to be trisomy or monosomy of chromosome, respectively. Such a situation leads to a very serious consequence in the individuals. Down syndrome or Turner syndrome, Klinefelter syndromes are the common example of chromosomal disorder, Down syndromes. This causes the genetic disorder in the presence of additional copy of chromosome number 21, trisomy 21. This disorder was first described by Langdon Brown in 1866 and affected individuals in a short stature with small round head and furrowed tongue and a partially open mouth. The palm is broader with the characteristic palm creases. Physical, psychomotor, and mental development is retarded. Klinefelter syndrome. This is a genetic disorder also caused due to the presence of additional copy of X chromosome, results into a karyotype of 47 XXY. Such an individual has overall masculine development. However, the female development of the breast, the gynecomastia is an expression. Such individuals are sterile. Turner syndrome. Such a disorder is caused due to the absence of one of the X chromosome, 45 with XO. Such females are sterile as ovaries are rudimentary besides other features including the lack of other secondary sexual characters. Summary. Genetics is a branch of biology which deals with the principle of inheritance and its practices. Progeny resembles appearance of morphological and physiological feature has attracted the attention of many biologists. Mendel was the first to study this phenomena systematically by the study of the pattern of inheritance in the P of the contrasting characters. Mendel proposed the principle of inheritance, which are today refers to as Mendel's law of inheritance. He proposed that the factor, later named as gene, regulated the characters of both pair allele. He observed that the expressions of the character of the offspring follows a definite pattern in different first generation, F1, second, F2, and so on. Thus, some characters are dominant and others over the other. The dominant character expressed when the factors are heterozygous condition, law of dominance, hence recessive characters only expressed in homozygous conditions. The character never blend in heterozygous conditions. A recessive character was not expressed in heterozygous condition may be expressed again when it is homozygous. Hence, the character segregates while the formation of gametes. Law of segregation. Not all the characters are shown due to dominance. Some characters show incomplete and some show codominance. When the Mendel studied the inheritance of two characters together, it was found the factor independent of a sort combine in all the premature and combination of law of independent assortment. Different combinations of gametes are theoretically represented in the square tabular known as Punnett square. The factor now as gene on the chromosome regulated the characters of a genotype and the physical expression of the character called phenotype. After knowing that genes are located in a chromosome, a good correlation was drawn between the Mendel's law, segregation, assortment of chromosome during meiosis. The Mendel's law was extended to form of chromosomal theory of inheritance. Later, it was found that Mendel's law of independent assortment does not hold true for the gene that were located on the same chromosome. These genes are called linked genes. Closely located genes assort together and distinctly located gene due to recombination assortment independently. Linkage and therefore linkage map therefore correspond to arrangement of gene on a chromosome. Many genes were linked in a sex and also called sex-linked genes. The two genes, male and females, were found to have a set chromosome which were common and another set of different. A chromosome which were different in two sexes were named as sex chromosome. The remaining sets was named as autosomes. In human, a normal female has 22 pairs of autosomes and A pair of X chromosome has XI, sex chromosome has XX. A male has 22 pair of autosomes and a pair of sex chromosome and XY. In the chicken, the sex chromosome is males are ZZ, a female is W. A mutation is defined as a change in the genetic material. A point mutation is a change in the single base pair in the DNA. A sickle cell anemia is caused due to change in one of the base in the gene coding of beta change of hemoglobin. Inheritance of mutation can be studied by the generating a pedigree of a family. Some mutation involves change in the whole sets of chromosome polyploidy or a change in the subsets of chromosome named aneuploidy. This helps to understanding the mutational basis of genetic disorder. Down syndrome is due to the trisomy of chromosome 21 where there is an extra copy of chromosome 21. Subsequently, the total number of chromosomes become 47. The Turner syndrome, one X chromosome is missing and the sex chromosome is XO and is studied in Klinefelter syndrome. The condition is XXY where it can be simply studied by the analysis of karyotype. Thank you.